the liquid media. But what is the difference between these two? The solid media has agar, 1.5, typically 1.5% agar. So the agar is the solidifier agent that we used it for solid media. So this one is the solid media, this one is the liquid media. And also in a solid media, as you can see, for the microbes in the environment, it was an agar plate. Agar plate is the solid media. The solid, you can see the colonies. Sometimes even you can count the colonies. But for liquid, it has the turbidity or cloudiness. So this is the translucent media. Actually, there is nothing in it. But sometimes when we grow the bacteria, when we inoculate the bacteria, it is look like this. This is the turbidity. The bacteria grows on that. You can see the difference between the translucent, no bacteria, and here is when bacteria grow. Okay, so what we are going today is we are going to inoculate the bacteria in a broth media. So how to tell if something is growing in liquid media? So the first thing is turbidity. Turbidity is the cloudiness, as I showed you before. But the other thing is pellicle. Pellicle is mass of cells floating, and sediment is a cell resting on the bottom. I will show you later what is the difference between these three. But for now, you should know that we have three different kinds of growth. So you already know that what is inoculum, what is inoculation. The aseptic technique is the practice of keeping your media from being contaminated. So you always need to follow the aseptic technique in order to avoid any contamination. And the loop sterilization. So in order to inoculate the bacteria, we need to use the loop. Here is how the loop looks like, but we need to sterilize it. Before each inoculation, you need to sterilize your loop. How? Here is the incinerator. We already turned it on, and you need to put your loop in incinerator at least for 30 seconds. 30 seconds to 1 minute is enough. So you can keep it for 30 seconds to 1 minute, and then we are going to inoculate 4 different TSY tubes. So our first sample is dirt samples. The other one is E. coli. The next one is Micrococcus luteus. And the last one is the sham. Sham is the negative controls. Just take this out. Sham is negative control. So we use the sham because we want to make sure that our experiment goes well. So typically on a sham, nothing should be grown because this is the negative control. So our lab manager provided for us these 4 different samples, and we are going to do the inoculation. But how should you do that? So I sterilize my loop. I will do it again. And here is Micrococcus luteus. Before you are going to do the inoculation, you need to shake it. Yes, because sometimes the bacteria aggregate on the bottom. In this case, you need to shake it well, like this. The same thing should happen for E. coli. Here is the E. coli. Okay, so we have the E. coli. Here is our dirt samples. And the sham, which is negative control, is actually the translocans media. Okay. Here is E. coli. And here is TSY broth. I will label it first. Okay. E. coli. My loop is still clean. Then you need to open the E. coli, which is provided by our lab manager. Grab some inoculum from it, and then do the inoculation, which means that transferring to a new environment where the bacteria can grow. Okay. That's it. Then again, you need to sterilize your loop because we are going to grab another different bacteria. So here is done. We need to put it in a coffee can, and then let me just show you where is the coffee can. Okay, here is the coffee can. We put our samples here. Then label another TS5 broth, Micrococcus luteus, M. luteus. The same thing for M. luteus. Here is the culture which provided by our lab managers. Our loop is sterilized. It's super hot. And then grab the inoculum like this. And then transfer to a new environment like this. That's it. Okay, that's cool. Then this is the next samples. We will put it in coffee can. Again, sterilize our loop. This time, we're the dirt samples. Dirt sample. It's important to label your tube, otherwise you won't know that what is your samples, what is this. Okay, this is already inoculated. I will touch the dirt samples. Try to grab a little bit. Okay. Inoculate in a tube, and that's it. Okay. Our last sample is sham, which is the negative control. Typically, we shouldn't do anything for sham. We shouldn't even open the cap. Otherwise, maybe the contamination happens. But we wanted to keep it super clean and sterilized. Here is the sham. We wanted to make sure that our experiment goes well. This is the reason why we use the negative control. These four already inoculated, and we will transfer it to the incubator. Again, 30-degree incubator. Like this. We have to keep it for 2 days in the incubator, and then we can see the results. So take out our cultures from incubators, and then we will observe the growth characteristics of the microorganism. The thing is that you should not vortex or mix the tubes, because we wanted to see the different type of growth. So here is the E. coli. You can see that there is a turbidity all over the tubes. Here is the M. luteus. I didn't shake or vortex the tubes. You can see the sediment on the bottom of the tube. This one is the soil samples. Again, you can see the turbidity all over the tubes and also some sediment on the bottom. And here is our negative control. It means that our experiments went well and we followed the aseptic technique. Nothing grew on our sham or negative control.